So our next speaker, first I just wanted to say, I think as most people in the room know, that immunotherapies are double-edged swords. So there was a little bait and switch here. So while Anna was going to talk about RIG-I agonists, she's actually going to talk about RIG-I antagonists, and her new company is Innatia, not RIG-active.
Thank you. So I'm very excited about having the opportunity to come here and tell you about a new idea that we have and to get your feedback. I'll be telling you about an approach to tune the reactivity of innate immune receptors as a sort of very upstream way to control targeted inflammation pathways. And so today we're going to be specifically talking about modulation of the RIG-I innate immune receptor. So RIG-I is a promising drug target for agonists certainly and antagonists as well. Just so you know a little bit about it, RIG-I is our first line of defense against infection by flu virus and flaviviruses and other things to detect and then respond to viral infection. But agonists of RIG-I are very promising agents for antiviral response, viral protection, and as part of an entity that Akiko Iwasaki and I are putting together, also for anti-tumor therapy. But just as Rob said, these kinds of things can be tuned with antagonists to help to control dysregulated inflammation that is directly a part of the RIG-I pathway. So let me tell you a little bit about the consequences of RIG-I hyperactivation, which is kind of a new area. RIG-I, when it binds RNA, elicits the interferon response and actually then turns on a relatively narrow set of cytokines. It is becoming clear that flu-associated COPD morbidity is due to RIG-I hyperactivation, Sjogren's syndrome and other forms of autoimmunity, acute radiation toxicity, and a lot of the toxicity associated with the new oligotherapeutic drugs are due to RIG-I. And so it's very nice to have a sort of RIG-I antidote, and that's something we're interested in. And this type of therapy is really new, so to just sort of benchmark it, I think the closest thing you can think of in terms of long-term value would be for a downstream cytokine. Look how successful Humira has been. The 2016 revenue was $15 billion. The market value for COPD alone in 2025 is expected to be $14 billion. So this could be a promising approach. This is grounded in real molecular understanding of RIG-I, which is, I think, an appealing thing. My lab solved the first crystal structures of RIG-I, caught in the act here of binding viral RNA. So we have a detailed molecular understanding of how RIG-I binds its ligands and induces a signaling pathway. We also created almost all of the in vitro systems for monitoring ligand binding and developed many cell-based assays for function. And this is an example to show you that we can come up with robust, sensitive assays for RIG-I activity. This is ATP's activity as a function of different RNA ligands. And so we have the sort of direct information from our biochemical approaches and structural approaches to develop good high-throughput assays. And we have a wonderful collaboration with Akiko's lab to then move this into animal systems to understand it in more depth. To embark on this, we did proof of concept studies with a curated pilot screen of 10,000 compounds. And in our first effort, we came up with an antagonist, just a hit, with an IC50 of 8 micromolar. Limited optimization in SAR resulted in a molecule we're going to call RIG-12 that had an IC50 of 0.7 micromolar with a very nice profile. And in a cell-based reporter assay of RIG-I signaling, you can see a dose-dependent response that's selective inhibition. So this sets the stage for large-scale HTS and lead optimization, which is what I'd like to do next. So using our assay platforms, structure-based design, and our collaboration with Dr. Iwasaki, we'd like to do a large-scale HTS on a very big number of compounds with an experienced CRO that specializes in this to the extent that preliminary SAR from the screening can be obtained from the screening data, and we can deduce which direction to go in for molecule design. I should tell you that the molecule I showed you on the previous slide had a molecular weight of around 300. We want to conduct limited lead optimization on promising hits and initial optimization of pharmacological properties. So what we're looking to do, because this is a very preliminary pitch for a very preliminary effort, is a seed series for about less than $1 million that would cover the HTS, CRO contracts, preliminary medchem, and also the engagement of experienced people to help us develop our idea. And so in this very early effort, I welcome any feedback, and thank you very much.
Questions from the audience? Carlo, no questions? Let me start.
So based on the biology of Rigai, to what extent do you need to inhibit it to see if there's a neurological meaningful effect in the disease?
So based on what we've seen, in terms, we've studied agonists of Rigai so that we have a pretty good idea of sort of how to induce a Rigai response, certainly in a rodent model, so that then we can then back titrate sort of with our antagonists and begin to answer those kinds of questions. In terms of a disease model, that would have to be induced with an agonist, to bring the inflammation level to an extent that's comparable to, the kind of inflammation response and cytokine response that you'd see, for example, in the Sjogren's model.
Okay, great. Thanks so much.